Namaste students, my name is JVS Dr. Neeraj Dholia, Faculty of Agriculture and Veterinary Science, Jayoti Vidyapeeth, Women's University, Jaipur. Today we are going to discuss a practical session, that is Eliza, and this belongs to the subject Medical Biotechnology Lab. So Eliza stands for the enzyme-linked immunosorbent assay. So this is basically an analytical technique, and it detects the presence of the antigens or the antibody for a specific pathogen. So this uses a solid phase type of the enzyme immunoassay to detect the presence of a ligand that is commonly a protein, but it may not be protein. Some other biomolecules may also be detected using the ELISA. So the ligand or a liquid sample using the antibodies that directed against the protein to be measured, ELISA has been used as a diagnostic tool in various fields, such as in the clinician for the quality control, as in the plant pathology, in the biotechnology. So it have various applications. So the very common type of ELISA, the antigens from the sample to be tested are attached to the surface. Okay. Then in this experiment, you need two types of the antibody. One matching antibody is applied over the surface so it can bind to the antigen. And you need a secondary antibody or second antibody with a detection system. So the one antibody is linked to the enzyme, second one, and the unbound antibodies are removed by the washing. So when you detect the presence of the ligand, the enzyme substrate reaction is happens there, and if there was finding the subsequent reaction for products, a detectable signal, most commonly it may be present in the form of a color, or other detection method can also be utilized. So to perform the ELISA, because this is a very specific detection, the sample with an unknown amount of antigen is immobilized on the solid support, and this is generally a polystyrene microtiter plate, or it may be other plates also. In the it have various types of the modifications. Okay, so you will aware of, or you will be studied for the types of the ELISA, that are the direct ELISA is there, the sandwich ELISA is there, competitive ELISA, reverse ELISA. So various types of the modifications are there. Okay, so of note, the ELISA can perform other forms of the ligand binding assays instead of the strictly immunoassays. Though the name carried the original immuno because of the common use in the clinician practices for the detection of the antigens or the antibodies, but there are modifications. Okay, so what is the principle behind the ELISA? So as we told that it is a biochemical test or biochemical assay. It involves the detection of one analyte. Analyte means the molecule which present you are going to study or detect. So that should be present in a liquid sample, and during the analysis, that stays liquid and remains inside a reaction chamber, or well needed to keep the reactants contained. So this is in contrast to dry lab techniques that use dry strips. Even if the sample is liquid, The final detection step in dry analysis involves the reading of a dried strip by a method such as the spectrometry or other methods. So in this method, Eliza separates some component of the analytical reaction mixture by absorbing certain compounds onto a solid phase, which is physically immobilized. So in Eliza, a liquid sample is added onto a stationary solid phase with a special binding properties. They have the binding affinity against the added sample, and this is followed by multiple liquid reagents that are subsequently added and incubated and washed, followed by some optical changes. So color may be changed or some other changes may be there. So in the final liquid in the well from which the quantity of the analyte is measured, the quantitative reading is usually based on the detection of intensity of transmitted light by spectrophotometry, which involves the quantitation of transmission of some specific wavelength of light through the liquid. So the analyte is also called the ligand because it will specifically bind or ligate to the detection agent or reagent. Thus, the Eliza it falls under the bigger category of the ligand binding assays. So, what are the various types of the Eliza? So, there are 4 or 5 types of the different Eliza methods are there that use the matching antibodies. Eliza tests are broken into several types of tests based on how the analytes and antibodies are bonded with each other and used. So the major types of the Eliza will be discussed here. One is the direct Eliza. So how to perform the direct Eliza? So a buffer solution of the antigen to be tested for is added to each well of a microgravity plate where it is given time to adhere to the plastic through the charge interactions. Then a solution of the non-reacting protein such as the BSA or casein protein is added to each well in order to cover any plastic surface in the well which remains the uncoated by the antigen. Okay, so this is known as the blocking. You are blocking the remaining space to prevent the unspecific binding of the antigen or antibodies. So the primary antibody with an attached conjugate enzyme is added and which binds specifically to the test antigen, coating the well. Then a substrate of this enzyme is added, and often this substrate changes color upon reaction with the enzyme. So the higher the concentration of the primary antibody present in the serum, the stronger the color change, and often a spectrophotometer or colorimeter is used to measure the amount or measure the intensity of the color or strength of the analyte present. Okay, so this was the direct Eliza. So now what is the sandwich Eliza? So because this is known as the sandwich, because here the protein is in between of 2 different types of the antibodies, or antigen is in between, like sandwich. So it detects this sample as antigen. So analyte is the antigen, and the steps are: first, prepare a surface is prepared to which a known quantity of captured antibody is bound. So on the solid surface, you will first attach the antibodies. Then a non-specific binding sites on the surface are blocked using a blocking buffer, casein or BSA or other proteins. Then the antigen coating sample is prepared, applied to the plate and captured by the antibody. The plate is then washed to remove the unbound antigens, and a specific antibody is added and binds through antigen, and the sandwich, the antigen is stuck between 2 antibodies. So the primary antibody could also be in the serum of donor to be tested for the reactivity towards the antigen. Enzyme linked secondary antibodies are then applied as the detection antibodies that also binds specifically to the antibodies Fc region. Then the plate is washed to remove the unbounded secondary antibodies, and a chemical is added to be converted by the enzyme into a color or fluorescent or electrochemical signals. So the absorbance or the fluorescence or the electrochemical signal of the plate wells is measured to determine the presence and the quantity of the antigen. Then in the competitive ELISA, a third use of the ELISA, that is through competitive binding. So how to perform this, and in this the unlabeled antibody is incubated in the presence of its antigen, that is in the sample. So these bound antibodies or antigens complexes are then added to an antigen coated well, and the plate is washed to remove the unbound antibodies. Then secondary antibody specific to the primary antibodies is added, and secondary antibody is coupled to the enzyme. Then a substrate is added, and remaining enzymes elicit a chromogenic or the fluorescent signal. So the reaction is stopped to prevent the eventual saturation of the signal. So this is the competitive ELISA. So next is the reverse ELISA. So this does not use the traditional wells. This test leaves antigen suspended in the test fluid. So the unlabeled antibody is incubated in the presence of the antigen that are present in the sample. A sufficient incubation period is provided to allow the antigen antibody reaction, and the sample is then passed to a scavenger container. This can be a test tube, or a specifically designed flow through channel. So the surface of the scavenger container or the channel has scavenger antibodies bound to it. So these can be identical or be sufficiently similar to the primary antigens that are free antibodies will bind. So the scavenger container must have sufficient surface area and sufficient time to allow the scavenger antibodies to bind to all the excess antibodies introduced into the sample. The sample that now contain tagged and the bound antibody is passed through a detector, and this device can be a flow cytometer or other device that eliminates the tags and registers the response. So these are the most common types of the variants of the ELISA, that is the reverse ELISA, the competitive ELISA, the sandwich ELISA, and the direct ELISA. So this is basically a biochemical test to detect the presence of an analyte, that is the analyte may be a antigen or an antibody, and the results are observed by the detection system either in the presence of a color, that is due to the enzyme link, which are enzymes that are linked in the secondary antibodies. So that was all about the ELISA. So hope you are satisfied with this digital session. If you have any doubt, any query, you can write in the comment box. And this session is powered by Digital Version 2, Jayoti Vidyapeeth, Women's University, Jaipur. Thank you.